afternoon, everyone. Good afternoon here in Düsseldorf. I know that somewhere all over the globe we have different time zones, so good morning, good evening, wherever you are joining. I am Lena. I am your guide to the 42+1 galaxy. And it's been only 4 months since we announced this pitch award and our DxPx to the world. And we are very excited and impressed about the resonance and the visibility that we already got. You all saw the numbers, 338 startups, all from our very own galaxy joining diagnostics, digital health, precision medicine, and life science tools. And we are very happy, of course, to have some helping hands. You saw that, our jurors, joining us physically today here, also joining us via a Zoom call. So we have some jurors in the back. You will see them later on. And yeah, of course, we want to thank all our investors who gave us the opportunity to present this pitch award of $2 million in funding to the startups that were submitting their pitches and presenting their companies to us throughout those 4 months. Today we will see the 8 finalists that were convincing our jurors the most, that were convincing them via their written pitches. And now we will see what they can do on a DxPx stage. We have 7 finalists joining us physically, and 1 finalist who is going to be our first finalist pitching today. He is calling in from the UK, ThinkSono. And ThinkSono is presented by Fouad Al-Noor, and they are enabling any healthcare professional to detect deep vein thrombosis at the point of care in only 5 minutes using an AI tool. And if there's nothing left, I would like to open up the stage for ThinkSono and Fouad Al-Noor. Thank you. There seem to be some technical difficulties. Perfect. Then this is your stage. I mean, of course, right now it's just only your projector, but your stage for the next 8 minutes, and after that we will hear 5-minute Q&A from our jurors. Have fun.
Usually has to be referred from their general practitioner to the emergency department, then they're risk assessed. Usually they have to have a blood test, and then they have to wait for the ultrasound appointment. Then they get a formal compression ultrasound scan, and then they get a diagnosis at the end. And that usually takes between 6 to 24 hours, depending on the institution, that entire pathway. In our case, if you implement our technology into the hospital, we expect that to take 15 minutes. And the reason is because now a non-expert can perform the scans, and that's really the key detail here, and therefore you can essentially skip a bunch of steps because it's much faster. You don't have to wait for that ultrasound specialist, and in some sense that blood test becomes less relevant. The reason they had to do all these other steps in the past is literally because there was no other way to assess patients prior to sending them to ultrasound. It wasn't cost effective, but with our technology, due to the ability of non-expert staff to perform scans, it's much more cost effective to do it the way we propose. And in fact, this has been validated by several different institutions. For example, we're working with Oxford University Hospital in the UK, hospitals in Sheffield in the UK. We're also working with St. George's. In Germany, we're working with Magdeburg Clinic, as well as Ernst von Bergmann Clinic in Potsdam. And we're also having conversations with Sana in terms of deployment. We're backed by the Wellcome Trust, as well as having ultrasound vendors such as Clios, who we can partner with. We're already CE marked as well. Our CE mark currently is about data collection and storage, and we're running clinical studies to make more clinical claims about our technology, for example, screening and diagnosis, etc. In fact, that's what the Wellcome Trust is funding, our multi-centered clinical study at Oxford, which allows us to make those claims later on and file for approval. We this isn't just research. We've actually published a paper in Nature on this, which shows we have very high sensitivity and specificity. For example, we have sensitivity shown above 90% and specificity above 80%, which is obviously very important for DVT. And so the future clinical studies, which we're currently running right now, is essentially adding on more to the evidence for us to file prior to filing for a CE marking. Now, to go into the economics of it a little bit, we also did a full-blown health economic analysis, which is also found in our Nature paper in detail if you want to look at that. But we can charge cost-effectively between $50,000 to $200,000, for example, per institution, depending on the size. And the reason those numbers might seem quite high is because the clinical pathway is so much lower. Another point as well is the way the go-to-market is that we have it's a pure software product, and we just charge a license fee per year per institution. So they just pay for the software depending on their size, and we have reoccurring revenue from that perspective. Obviously, we worked this out that this makes sense for the hospitals too, and I'm more than happy to take questions on the details of this. I think I should probably go into the team as well right before I end this. Obviously, it's not just myself. We have my co-founder Sven, who's our CTO and essentially architect of the product, but we also have machine learning engineers, radiologists, hematologists, vascular surgeons, and other commercialization advisors who help us to commercialize in the next stage of the company. In fact, that is literally the stage we're at now, where we're planning to raise a Series A to commercialize the technology further once we get our approvals as discussed. I'm happy to take your questions.
Could you elaborate a little bit more on evaluating the data? How do you make sure that the users, clinicians arrive at the correct assessment? Mm-hmm.
That's actually the core IP as well. So the way we do it is in our study, we compare ourselves to the standard of care. So you have, so you can imagine, right? So you have a suspected patient coming in and no one knows if they have DVT or not. They're scanned in our, let's call it ThinkSono pathway. So they go through a nurse or a doctor using our technology, scanning them. And then the same patient blinded is going through the standard of care. So that's their radiologist or sonographer, whoever usually scans that patient. And we only find out at the end of that process, we compare those answers directly. So we really want to see, is there any difference between our sort of clinical pathway and the current clinical pathway? And that's the way we've essentially done it. In fact, we're already getting literally evidence as we speak from Magdeburg running exactly this study. The study in Nature that we just published in Nature, that was retrospective. So we knew retrospectively, at least the initial clinical evidence is strong, but obviously we want to build up more clinical evidence to make sure that there is no sort of adverse events by going through this much quicker, much more simple pathway relative to the current one. We don't plan to be better yet, although I can argue later on, we may be, but we aim to be equivalent, but obviously more cost effective, better patient outcomes, faster, etc., and more accessible. You can go to remote areas and scan patients. You can scan many more inpatients. So there are many, many, many advantages that compound. But the first step is making sure that we're not worse than the current pathway. We're at least as good. That's what we're trying to claim.
Sounds great.
In your pathway, who's actually using the device? Is it a nurse or still a physician? Because, I mean, you obviously do the picture analysis via AI, but you still need to have the right place where you actually place the ultrasound device. Yeah. So you have to be trained to use it, even if there's an AI that's actually analyzing the pictures.
Yeah, so that's a very good point. In different jurisdictions, it's slightly different. In the UK, nurses are seen slightly differently than in Germany, for example, so they actually perform the scan. But in fact, it's almost always, I call it the attending clinician or the first point of care. Sometimes that's a nurse, sometimes that's a doctor. So in some sense, the whole point of the AI, it's operator independent in some levels, so it doesn't really matter who does the scan. There is a review, if you note that I said there's a review phase, that is still done by an expert, and we call that Auto DVT version 1, because we don't want the decision making to be done by the nurse. We still want there to be a check. The economic argument still holds very well because they don't have to scan them themselves, so therefore they spend 1 minute reviewing as opposed to 20 to 40 minutes scanning and taking in the patient and coming into the hospital. So the first phase is scan typically done by the nurse or by the attending doctor, who doesn't know ultrasound, obviously, in that case. Otherwise, they can diagnose them themselves. But the non-expert operator performs a scan, and then there's a review phase, and then you get essentially your assessment, your screening. And then as we build evidence more and more, you can imagine how you can do this. If you run a larger study, for example, after this study, we plan in the future to do what's called a non-inferiority study. And then we can start to make even larger claims. I believe we would need probably 3,500 patients, and it's quite big with multiple centers and stuff to really show that this is safe. That, for example, a nurse can just screen the patient and not, there's no decision to be made after that. But for now, we always have that double check at the end, just to make sure. Yeah.
Do you already have a pilot customer with whom you can show that the business model can be run profitably?
That's exactly the phase we're at now. So we just this year, we reached a point where we were able to deploy our technology onto the app, as I described, this Android app, which wasn't possible for us before. Technically it wasn't possible. Now it is possible. And we have Magdeburg, RWB, Oxford, Sheffield, St. George's, another hospital in the UK called GTD Healthcare, and we're expanding to the US. All of those customers are piloting the technology either via a study or via slightly more commercial terms. But the idea is to convert, as you could imagine, we want to convert all of them into customers at the end. So we're talking at least 5 to 10 potential customers already. But we don't have contracts yet. And that's very simple. You can't sell a technology you haven't proven. So we're not that naive.
Yeah, but do you have some sort of a plan, which kind of performance you need to show until we get a contract with them? So if you show results X, Y, Z, you will get a contract?
Yeah. So we have to at least show, based on letters of intent and other information we've got, and obviously speaking to CEOs of these hospitals, you have to show cost effectiveness and that the sensitivity at least high enough. Specifically, the sensitivity has to be high enough because you can't miss the clots, right? Then you're causing a bigger problem than you're trying to solve. But the health economic argument, although we've done the entire modeling, that's still not good enough. You need to do it in the institution and show as a reference site that this is true. And that's exactly what we're doing at RWB now. That's what we're starting to do. So we're building up, we've been working on making sure that, let's say, the clinical stuff is done. And then now we're shifting towards the commercialization stuff, which is the models that we built actually true? That's basically what we're doing.
Okay, then maybe one thing for me, Düsseldorf speaking again. Hi, coming from the off. We do have some questions from our on-site jurors. I will first give the microphone to André Zimmermann, SHS Capital.
Yeah, thanks for the presentation, really excellent. Maybe can you explain a little bit more about why you think you can charge 50 to 200K?
Yeah, sure. So we went to Sheffield University, and they specialize in health economics, at least for the NHS. I know that's not quite Germany, but they're not so drastically different actually in terms of scan costs. So let's assume if you give me that grant that it's going to be somehow on the same order. What they did was they worked out 6 different ways you can implement, literally like slot in our technology, and they call them algorithms. And so when you slot it in, you can work out, okay, how many D-dimer tests do you have to do? How many scans do you have to do? How much does a scan cost today versus how much our technology will cost? They also worked out based on our accuracy. So, you can, if your specificity, if you have too many false positives, it becomes less and less useful, as you can imagine, because you're sending everyone, for example, for a formal scan. So based on this model, they worked out that we can charge as much as £174 per scan. If you work it out per scan, you wouldn't charge per scan, obviously. But if you work it out per scan, they worked out, I believe, £174. And again, you can find the details in our Nature paper. So it's all peer reviewed. If you just Google DVT AI, it's one of the first things that come up, and obviously I'm happy to share the link as well. So that's essentially what we did. Yeah.
I'm sorry, I need to stop you here because we want to hear the other finalists too. And there's still some question from Angus McKilkin from McDermott.
I just have a quick question. I'm curious, how much has been raised so far and where has it come from?
Okay. We've raised about £3 million sterling. So that's based on venture capital funding and some funding, about 400,000 roughly, from the Wellcome Trust for our clinical trial. That's how much we've raised this much. I'm not going to say exactly how much we're going to raise for our Series A, which I might imagine that's the next question. And the only reason is I would rather discuss that more in person because there's more negotiation to be done about exactly how much we need. But we're planning obviously to raise more than that for our Series A. That's going to be a substantial round.
I saw that Jens Baas from TK raised his hand. Is this a quick question? Because otherwise I would give up to the next finalist.
It actually is. How big is the database for your AI training, and where's the data coming from?
We spent 5 years collecting our own proprietary data set, not because it's fun, but mainly because there's no data set for compression ultrasound pretty much that you can do for DVT in particular. So we have, I mean, I don't know if this is a controversial statement, probably the largest data set in the world for curated DVT compression ultrasound data. The way we collected it was via initially healthy people, because you need to first get basic anatomy essentially. And then we did a data collection study at Oxford University Hospital, as well as Ernst von Bergmann Clinic, to further improve the algorithm. And then we were satisfied, that's what we based the paper on actually, that actually this is good enough, at least to run in a bigger clinical study. So the answer is a mixture between healthy people, healthy volunteers, and patients as well.
Okay, with that answer, I hope our jurors have all the information they need. And I would thank Fouad for your presentation and for answering all those questions. And I would like to welcome the next finalist on our stage. We are going to see Fertigo Medical as a second finalist pitching. Tsafrir Kolatt came here yesterday from Israel, and he's going to present us his company who helps increasing the success rates of IVF treatments with continuous real-time in vivo monitoring. Your stage, have fun.
Good afternoon, everybody. First of all, let me say that I'm honored and proud to be one of the 8 samurais that made it to the final presentation. But the main thing today is to present Fertigo Medical. So Fertigo Medical started with a couple who were trying to conceive through IVF with no success. So then we joined them, a group of well-experienced scientists and businessmen and medical doctors. You can see there, some of them are listed here. And we thought of a solution, whereas the need was very clear from the very beginning. Only about 30% of IVF, in vitro fertilization cycles, succeed. And this imposes a huge financial, mental, and physical burden on the patients and the families that undergo IVF. We realized that there was a blind spot in this process, and that was the timing when the fertilized eggs, namely the embryos, should be introduced back into the uterus. While the biological time frame is between 12 to 36 hours, modern medicine currently know the window of opportunity to an accuracy of between 3 to 5 days. And this mismatch is responsible for many failures of IVF cycles. We realized that as soon as we come up with a solution to this difficulty, it will immediately catch up simply because it is a very competitive market. And indeed, it took us a few years of research and development, and it is not only that we came with a solution, but we actually came up with a system to implement this solution, which is not less difficult. So Fertigo Medical introduces an evidence-based, namely scientific, solution of a real-time, in the current cycle, in vivo monitoring of the uterus readiness to implantation. Eventually, it should lead the physicians to aim at the right moment when they put in the embryos. We do it by looking at the endometrium, which is the inner lining of the uterus. Now, the endometrium is the fastest growing tissue in the human body. Faster than our nails, faster than our hair. It grows for about 0.5 millimeter per day, and of course, after 28 days, on average, sheds out. So, however, it is not only thickening each and every day, it also entirely changes its inner structure, its morphology. And this is what we are after. So we are looking at the evolution, the rapid evolution of the endometrium, of this inner lining of the endometrium. And this is a clear evidence for the readiness of the uterus in the current cycle to accept, to receive the fertilized eggs. So what we are doing, we are introducing a mini probe into the lower end of the uterus, as if we had a microscope, a tiny microscope, put into the uterus, looking through a magnifying glass at the structure, the finest structure of the tissue itself. So we take images of this fine structure, and then we send them off to analysis. Of course, AI-based analysis, among other things, among traditional image analysis, but not only image analysis, but also time analysis. And therefore, we're able to take a handpiece, which is a multi-use handpiece. This is the actual device that we made in order to supply this purpose. So the multi-use handpiece basically controls the hysteroscopy session. It is engaged to a single-use disposable device on the left-hand side that looks at you. It's a small camera that looks sideways to the walls of the uterus, just like a microscope. We had to anchor it to the wall. And then, as soon as the handpiece captures an image, it is sent to our AI-based computational unit in order to get analyzed. Everything is engulfed within one in-all state-of-the-art software that does not only do the previous things that I mentioned, but also stores all the data so that we can compare different days of the same cycle, different cycles, same days of other days, and more importantly, we can compare it with standard libraries to see for the patient when she is on her cycle. Of course, we had to validate it, so we went through preclinical validation trials with very nice results of correlation for what we call endometrial dating. And then, after the device and the system were ready, we started also real safety and usability, and this is the first time ever that you see in vivo uterus from within. All the fine details of blood vessels and outlets of the glands. So this has unprecedented resolution, and it basically provides us with a tiny microscope within the uterus, telling us through the algorithm how well we can aim at the right day. We went on to human clinical trials with ex vivo samples, and again, we found a very nice correlation with traditional gold standards, such as testimony of the patient, or if you wish, blindly assessment of pathologists for endometrial dating, or hormones in the blood. So, all in all, the main issue is that the benefit is it is non-invasive. We do not need to collect anything. We do not need to take samples. We simply take pictures. It is evidence-based, so it is not based on any gut feelings or assessments or art or witchcraft. We have high accuracy of plus/minus 12 hours. It is complementary to all other IVF treatment forms. Well, I'm not going to tell you about the booming women's health market, which has been long neglected and really deserves our attention now, but just a quick glance, well, never mind. You know the numbers. It is a huge market. It is a really huge market, the IVF, just multiplying the number of cycles and the cost per cycle gives you more than $15 billion per year only in Europe, here in the United States, and in Japan. In a year or so, we are going to have our CE and FDA cleared for the hysteroscope, and a year later we are going to have our IFU, namely increased success rate. We couldn't find a better partner to team up with than EVRMA, which is the largest fertility clinics network worldwide. Here in Europe, it has 65 clinics. It originated in Spain. Now it has also clinics in the United States and in Latin America. They're doing with us all the clinical work and all the regulatory clearance. I'm not going to go through all the benefits of investing in Fertigo. I would like to point out one issue. Last August, our main competitor was acquired for €1.25 billion. It's an enormous sum, and they're doing a very elegant RNA test for the same thing we are doing. As a matter of fact, we are only two companies and only two different approaches. We are doing it in vivo, so we can do it real time. And we can do it today, tomorrow, and the day after tomorrow. And then we have a new dimension to pathology, the time dimension, that has not been seen before simply because it was not useful for any other tissue other than the endometrium. We can do it endless times, and we can do it, this virtual biopsy, time and again in different cycles. So, you know, the couple from the first act are now parents of 2 twin boys, so we can sum up after that. The main thing that I'd like to say is that modern medicine should really now be personalized and should really now aim at all the tools that we have at hand, be it artificial intelligence or be it big data approach. Putting it all together, it can become an evidence-based medicine. This is what we are after. It took us a long time to convince the medical community that indeed we are on the same train, and we couldn't do it without our partners, but we won't be able to do it without your help too. Thank you very much.
Thank you, Tsafrir. Maybe we can start this round with our jurors on site. Erika? Yes. I'm just going to give you the microphone. So Erika Stanzer from McKinsey.
So how would this work in terms of the kind of clinical treatment paradigm for someone who's going through IVF? Would they have to now come in like every single day, or like how does it fit in with the timing and treatments that someone would already be receiving?
The nice thing about IVF is that there are already scheduled visits during the critical days. The critical days are between the ovulation and day 14 in general standard and day 21. So they are undergoing ultrasound anyway in order to see whether the endometrium is thick enough. This is a threshold criterion. So we are going to use the same scheduled visits, and our procedure takes about 5 minutes the most, because we basically enslaved an existing shaft pipelle in order to put in our tiny camera and to picture the endometrium wall.
Thank you. We do have another question from Angus.
I'm curious how much has been raised so far and where did it come from?
Sorry, I didn't get it.
How much has been raised thus far and where did it come from? How much money has been raised so far for the company and where did it come from?
Thanks. We raised about $3.5 million. They came from the government of Israel, which was a nice kosher stamp for us because they are very tough. So it is Innovation Authority that gave us a grant, non-equity grant, of course. It came from an Israeli VC. It came from individual physicians, the most celebrated IVF experts in the world. It came from CLA, from EVRMA, who really believe in this. By the way, our competitor, iGenomics, also emerged from EVRMA. So they know something about good technology.
Thank you.
Good, thank you. Next on we have Bernd Görgen, HTGF.
Yeah, thank you very much. How much would you expect is the success rate of the IVF procedure increasing? What's your design goal regarding that? And how much are you going to charge for your system? And to what extent is that translating in an increase of cost for the IVF procedure?
It's a nice question because the two parts of it are well connected, you know. As well as if we can improve it by a lot, say 50%, then of course we can charge more. But we are very modest at first. We think that it is going to be a few, some like up to 10%, which is unheard of in terms of success. I mean 10% more, unheard of in terms of increasing the success rate. And right now, it depends on the territory how much we are going to charge. It is going to be about 1/10 of the cost of an entire cycle, because we are going to spare a few cycles. So that brings us to, figuratively speaking, about $1,000 per package deal. Namely, not only the disposable, but also the analysis of it until the final answer.
Okay, and then we have another question.
I understood that it's translating, it's $1,000 to $2,000 for an increase in efficiency of 10%, right?
Yes.
Okay, thank you.
Sure.
Okay, thank you. Jens Baas, TK, last question.
You mentioned you're also doing a longitudinal study or longitudinal looks at the endometrium. Are you not worried about infection? How long is that device staying in place? I mean, if it's staying in place for a couple of days or even weeks, I as a physician, would be worried about infection there.
Well, you know, our vision at the beginning was that it will stay there for 3 days, but no longer. Now we simply treat it as a hysteroscope. So we put it in for a few minutes, 1 to 5 minutes. We take pictures, that's enough, we take it out. And this saves a lot of headaches, regulatory pathway, and so on and so forth.
Thank you, Fouad.
Thank you.
Next up, we have our first German startup, Dermagnostix. Also live on stage here today is Anja. Sorry.
Natalie. Do you have the pointer?
The pointer is on the table?
It's on the table, okay.
Yes, it's on the table. So, and Dermagnostix is developing the first fully automated, cost-efficient, microfluidics-based, it's a long sentence, portable lab-in-a-box for molecular diagnostics in dermatology. And the translation is coming now.
Yeah, hello everybody. Thank you so much for inviting us today. We are super proud and happy to be here and get this incredible chance. To give a broad picture, at Dermagnostix, we are moving diagnostic frontiers, nothing less is our vision. And with skin diseases, we really target highly relevant and prevalent diseases. 1/3 of the world population suffers from at least one skin disease. The positive news is that over the last decade, so quite recently, many specific and highly effective therapies have been brought to the market with which we can treat our patients better than ever before. However, these treatments, and they're quite expensive, so if you go for atopic dermatitis, up to 20,000 euros per year and patient, all these therapeutics are administered based on a diagnostic system that has barely changed over the last century. It's based on clinical view and histology, and that's super imprecise and subjective, and a lot of questions still remain unanswered. So for some entities, up to 50% of patients are misdiagnosed when it comes, for example, to hand and foot areas. Individual course of disease, for example, in melanoma, cannot be predicted, and even if we apply the best therapeutics available for atopic dermatitis, only 30% of these cases show remission, and we cannot predict who it is. And the only discipline that can really provide answers to these questions is molecular diagnostics. And it's not just us that we see it coming from the academia wanting to really make this company happen. It's also industry, pharmaceutical companies that see that with companion diagnostics, guided clinical trials, they could save billions of dollars. And also healthcare systems could save a lot of money if doctors applied those therapeutics based on objective markers instead of trial and error, which is currently the case. And also, according to our survey, 80% of dermatologists see that their diagnostics is not perfect, and they would offer molecular diagnostics if technical feasibility is given, and that's exactly the problem. Manual implementation of these complex molecular assays is super difficult. Maybe at this point I should explain what is complex assays. We're not talking about measuring one single bug in a swab, which is the case with COVID testing. We're talking about the test of today and in the future, which are complex gene expression-based markers, several markers on gene expression levels from solid tissue, everything challenging by itself. And all this needs to be implemented. And if you do it manually in your laboratory, it's time-consuming, several hours up to days. It's hampered by human errors. If one does it today and the other one does it tomorrow, there are differences that we cannot tolerate in these settings. And it requires a lot of resources and high standards. Taking all this together leads to the fact that we're not having a broad implementation of molecular diagnostics. Even if we have really great publications out there, they are ending at the stage of publication, not making their way to the patient. And this is now the solution we offer, a patented automated mini lab for a paradigm shift really in skin diagnosis. The system consists of the instrument, the lab disk analyzer, and the lab disk. I just brought here one sample. The lab disk looks like a CD. You only need to put a part of your skin sample, it could be a biopsy, I'll show it later on, close the lid of this cartridge, insert the lab disk into the lab disk player like a CD in a CD player, and all processes, analytical processes, from the RNA extraction to the PCR at the end are performed fully automated. We cannot only deal with histology blocks, mini biopsies or blood. The real paradigm shift is that you can also deal with tape strips. So it's a non-invasive sticker. A dermatology directly at the patient site can stick to the patient's skin. It's non-invasive, non-painful at all, can directly put it in the machine and 1 hour later can tell the patient which therapy to take, because we can measure just by the epidermal layers with a few squamata actually on the sticker, we can measure complex gene expression signatures. Our system is precise, objective, standardized. It's a closed system. We can measure 40 targets and more. And why am I saying this? Because we are able to adjust our cartridges to the assay and not the assay to the cartridge, which is usually the case in other systems. That means our system is super simple, very simple physics, rotation and temperature. We don't need filters, pumps, we don't generate bubbles. We can actually generate a first model on a foil within 1 week. And only if it works on this really cheap foil, we then go to the injection molded cartridge, which is a huge advantage in terms of cost-efficient and rapid production developmental cycles. And last but not least, it's easy to handle. Everyone can do it. Even someone who doesn't understand what molecular diagnostics is, because it's complicated, can do it right away in his practice by just using our device. Already with the first 3 products, we are addressing a very immense market potential. The first test is Sorex, our first patented molecular test to differentiate psoriasis and eczema more precisely than current standard diagnostic does this. It's actually the first gene expression-based test anyhow in inflammation, not only in oncology. It's already reimbursed by a German healthcare insurance company, and we're having industrial trial with Leo, where it exactly comes to the point, filtering out patients before giving a therapy. Next product is MutationX, where we are targeting all therapy-relevant tumor mutations, not only in skin melanoma, but also beyond in other tumor entities. And it's fully reimbursed, several hundred euros already in Germany. That shows that here we are talking really about attractive reimbursement scenarios. The third product is Lymphlex, a very important product because here we differentiate malignant tumor, skin tumor, from benign inflammation. Why is it important? Because at a very early stage, the skin lymphoma can be treated, they're curable. But later on, patients are palliative cases. And in this early stage, current diagnostics cannot prove really good diagnostics, and that is actually, this gap is actually filled with our product. To really continuously fill our pipeline, both biomarkers and technology, we focus on R&D, microfluidics, and biomarker development. We work together with partners for the lab disk analyzer, the lab disk, whereas we do the functionalization in-house with our shareholder, Hans Schickhardt, who is world-leading Institute for Microfluidic Systems. In Germany, we can build up our own distribution channel. Why? Because few pathologists digest a huge sample volume. So already by targeting few large centers, we can generate already very good income in the first year when we are on the market. Whereas in the US and in Europe, we have to work together with distribution partners. Concerning the customers, it's pathology labs when it comes to histology blocks. But when you think of stickers, we are already at the patient bedside. We are at dermatologists. We can even be at pharmacists, and really have a broad circle of customers. We're generating our money according to the razor blade model, because here with our cartridges we can produce them at a very cheap price and generate attractive margins with those. Who is the team? We're an interdisciplinary team of well-known, I would say even key opinion leaders in the field of dermatology, biology, AI, and microfluidic engineers. And so from the first idea of a new test on the dermatology side, we are generating new biomarkers with our AI help, getting new assays to work and bring those assays onto the cartridge. So the full pipeline actually is mirrored in our founding team. And when we come to the roadmap, where we have raised so far almost 3 million euros of highly competitive third-party funds plus pre-seed funding money to get actually this company started. Amongst that, an ERC starting grant, ERC proof of concept grant. We are now about to finalize a seed round of 2 million euros, and the really cool thing for us is that our seed investors are pathologists, dermatologists, because they saw the need and they said right away we want to invest in that because we really need it for our daily practice. And now we're about to raise another one, Series A1 and 2, to get the product ready and get it into the market. Thank you very much for your attention, and I'm happy to answer questions.
Thank you very much, Natalie. Glad you could join. We heard you had some train troubles in this morning, so happy to have you here. Are there questions from our jurors? Yes, Angus.
Since you already answered the question of how much money you've raised and where it came from, I'll ask a different question. Looks like reimbursement is figured out in the German market. What is your timing and what are your plans for other market entry and securing reimbursement for other markets?
Yeah, here definitely we are having, it's still under NDA, I cannot talk about it, but we have a very interesting cooperation with a US-American partner that have already a test on the market that refers to the technology that we have. And here we are seeking co-development, so kind of providing our platform for their application so that we can get into that reimbursement system. And definitely we have to work together with regulatory experts when it comes to the approval in other countries. And we have also been quite happy to be selected for the CDL laboratories in Oxford to get also more knowledge on how it's reimbursed in other countries of Europe, because it's absolutely sure reimbursement is a key to success in diagnostics. But we are very confident because some gene expression tests are out there that are already reimbursed. We didn't believe it for 3,000 euros in Europe. And when you see the prices for what we can produce, I think there's quite a good chance that even if it's lower, we have attractive margins for us and our customers, which are the dermatologists and pathologists.
Thank you. We have a question from Jens Baas.
Well, you're currently focusing on dermatology, which is probably a very good starting point, but is there any technical reason why that shouldn't work in other areas?
Yeah, we're not just in dermatology. For the mutation X disc that we are working on, we are not targeting melanoma only, but we are targeting all the mutations that are not only prevalent in melanoma, but also in other tumors where the same mutations occur and where the same therapies are prevalent. So that it comes from dermatology is our interest. I'm a dermatologist. We have been working on the test, published it in 2014. New England Journal of Medicine followed another cool publication, and we saw, okay, we cannot come further with exactly the problems, and that's why we see this market completely underdeveloped. The second point is that in no other discipline in medicine so much has happened over the last years. I mean, we have from no therapy 10 years ago, we have now more than a dozen of therapies already available for psoriasis. And now we're here in our clinical practice having those 15 biologicals, not knowing which to apply in the patient. And that's, I think, the hugest reason for it. There's a lot of money, there's a full pipeline in dermatology, and this subject is going to be transformed completely from a pure visual discipline into a completely molecular discipline. The complete career is going to change for dermatologists. There have been people just creaming and maybe doing an excision, and now they have to become immunologists, understanding what they're doing with the therapy, and that's why it's such an interesting field to start in dermatology.
Thank you. We have another question from Andre Zimmermann.
Yeah, thanks for the presentation. I have two questions. One is, do you have to do all the 40 tests within your disc for every patient? Because I think if you have a skin cancer, it's different than when you have other skin diseases. That's the first question. And the second question is it practical that the patient has to wait 1 hour for the results? So that the dermatologist has to keep the patient for 1 hour in his practice?
So for the second question, maybe I start with that. It's the best what can happen to a patient if he gets a result in 1 hour. The alternative would be not getting a result at all, going home with the wrong therapy, and being lucky enough to have a second appointment at the dermatologist 6 months later. That's currently the situation in dermatology. And in that level of precision, there is no diagnostics out at all. So this is, especially with the sticker technology, it's a paradigm shift, and every dermatologist would see that 1 hour is nothing. For the mutation diagnosis that we're currently doing, some people wait several weeks. In the US, 25% of patients are started actually on a €100,000 melanoma therapy without knowing what mutations they have. So that's the current status, actually, the poorness of diagnostics in the market. The second one is concerning the 40 tests. So each test as such is a closed disk, and this test can consist of several markers. So you only apply the test of the test portfolio that applies to the disease. So if a melanoma patient comes in, you use the melanoma, the mutation X disc, and not the other one. And within the mutation X disc, there you have the 40 markers. And yes, you need it because there's not just one single mutation. There are heaps of mutations. All of them have different sequences that you need to mirror. And in addition, I did not make this clear yet because it's still confidential, but we're not only measuring tumor mutations, we're also looking for markers to predict actually tumor survival in melanoma and predict what is the best therapy option actually for a very early stage melanoma case patient. And then 40 markers are not even enough. We have to increase our disk, and that's the advantage because you can easily do it just adding some more channels in the disk and not throwing away the whole cartridge and making a complete new cartridge, what would be the case in other companies.
Okay, and we have a final question from Bernd Görgen, HTGF.
Hey, Natalie, thank you very much for the presentation. My question relates to the putative medical value of your diagnostic system, meaning that in which of the listed diseases or indications you gave in your presentation is it absolutely critical to get the result in the doctor's office a couple of hours earlier than from the central lab? And the second part of my question is, in which of these indications would you immediately, after having received your result, start with the therapy? Immediately after, not a couple of days or weeks later.
It's true, we are not that time critical as, for example, in infectious diseases on the intensive care unit where you have to act immediately. It's about seconds and minutes that you have to take care. But it's still an advantage, for example, in psoriasis patients, when they come to your practice, before you're right, a steroid cream would have been enough, and then they come again. But now, actually, when they have a disease core that is heavy, you can start directly, and that's first-line therapy with a biological therapy, and you have to start it right away, because we know now that it's not just a skin disease. No, it's actually a systemic disease that can affect your, that can cause arthritis, that can also affect your circulation system if you do not treat it early enough. The second thing is that it's not only about time, it's about precision. And that's why I said the manual performance of this assay is not possible at all. We started with it, we developed our first test, we published it, we got a lot of samples from clinics outside who said, can you not just process it? And we said, yes, we can, because we have the experience and we have the expertise, but we cannot really scale it up. We cannot bring it in other countries, because all of them would have to go the story that actually we went through. Building up a lab, bringing up the expertise, knowing the protocols, how to do it. And that's actually the most decisive point, to have the precision, and that's only possible in a closed system. The extra time that you can save is an extra on, but it's not the only reason for our system.
Can I have some? Thank you. All right, thank you very much. I think we are, yes, okay. Then we're going on to our next finalist. Next up, someone you probably already saw when you are following our YouTube channels, or if you have been in the loop with our US satellite event this year in July. We have Marc from Alertgy joining from the US, and he tells us today a little bit more about his on-demand, real-time blood glucose monitoring in the convenience of a wristband.
Guten Tag und danke, dass ich hier bin. I have to practice my German, so thank you very much for giving me the honor to come here and talk to you today and tell you about Alertgy. I started Alertgy several years ago because my wife, one morning, almost went into a low blood sugar induced coma. Back then, there was nothing that you could have, that you could buy, that would warn you if your loved one was in danger. Fortunately, I was there, and I noticed the condition, and I took the right actions and saved her life. You know, she woke up from her stupor, and she said, "Where am I?" I said, "You're at home, and everything's fine." But you know what? It wasn't fine. And I thought, what if I would have not been there? So that started my journey. So our mission is to save lives, is to improve the quality of life of diabetics, and also to prevent 2 billion people on this planet that are borderline diabetic from becoming diabetic. Did I turn it off? Again, it's a major problem. In the world, there are over 500 million diabetics, with 100 million people that don't know they have the disease. They're a walking time bomb. I myself was a type 2 diabetic, and I saved my own life. So no matter what happens with my company, I'm ahead because I'm alive and I'm here today. When I found out I had diabetes, it was with my own system, and my blood sugar was 500, and my A1C was 12.8. When I called my doctor and I said, "I'm diabetic," he said, "Well, you're not a doctor. You can't diagnose yourself. Come into the office." And he tested me. He said, "I'm sorry, Mr. Rippen, you're diabetic." I said, "Yeah, no kidding." But you know, I should have known that because it ran in my family. But I thought, because I had just lost about 80 pounds, that I was in the best shape ever. I could wear my college clothes. But it's because I was hyperglycemic and my body was actually reacting with my fat and my protein because it couldn't use glucose. So it's a tremendous problem, and it's growing every single day. We graduated from the Cleveland Clinic Health Tech Accelerator, which is part of a plug-and-play program, which I've graduated from several plug-and-play programs throughout the world. The chief of staff sat down with me and said, "Marc, this is the most critical problem we have in our hospital system today. 1/3 of all the people that come into my hospital emergency room, they're being admitted because that go into the hospital are being admitted because they're diabetics that have lost control over the disease. They refuse to prick themselves. They refuse to see what they're doing. And so their doctors don't have any data. And they stay in my hospital for at least a week. Some 2. They come in, they have a stroke, they have limbs that are being amputated, they're having organ failures. This could all be prevented if we could just give them a device that they could put on their wrist, that they don't have to think about, that if they want to know, we'll tell them. If they're in trouble, we'll let them know, let their loved ones know, and if necessary, get emergency services to them to make sure that they're okay. The technology is not new. It's been around for decades. When I was a director of engineering at Stanford Research Institute in the Marine and Space Technology Group, my main mission was to take technologies developed at universities and government research labs and to apply them to real problems that the military, DARPA, special operations, had to solve. So my specialty is taking technology and making it practical and demonstrating its application in the real world. I had been working on electromagnetic sensing technology for military applications and shallow seawater applications. It turns out the very thing that I've been spending the last 6 years on, I had the answer to the question that no one had before, is how do I make a sensor that can allow me to look into frequencies that we need to look for deep inside to the body using RF energy. And I built the first device and it worked. I had never heard of dielectric or RF impedance spectroscopy. I'm not a physicist, but I am a spectroscopist, and I knew molecular spectroscopy was not the way to go. I had worked with Nicolay for a period of time, and infrared and Raman is great, but it's not going to give you the results you need for what we need. We penetrate deep into the body, so we analyze the glucose in the blood, not interstitially. Every single other continuous glucose monitoring technology that's approved today, and it's going to be approved in the future, is not capable of measuring glucose in the blood. And so they're always never going to be real time. We're real time. And I discovered, as we were going through the spectrum, that we can also see insulin, which is another whole interesting thing to look at as we go down the road. So really, it's a tremendous opportunity to do a lot of good in the world, to get this out there so that people can actually have a device that they can just wear on their wrist. And the beautiful thing about our device is that it is designed to be able to be put onto a smartwatch or just a wristband. And this is just the beta prototype. We're going to be making this even smaller. The first device that we tested in clinical trials was actually the size of a Motorola. We call it a brick. The market is perfect because we have digital health, we have wearables, IoT, AI, everything's all come together, and this convergence is going to help us drive it to market very quickly. We're looking to raise $10 million to get us through regulatory trials here in Germany, as well as in the United States, to also develop the next generation to make this even smaller and more power efficient, and to move the product forward as we go down the road. We have two products. We have a clinical version that was tested. We've proven it works as well as best in class at the Cleveland Clinic, as well as another trial, which is peer-reviewed, both are peer-reviewed journals. We have the AI fully developed, the Google Cloud platform, and scalable. And we have data to show that it works good enough. And I believe as we get more and more data, it'll work even better. We have something that lasts years, not days. We have something that's non-invasive. You don't have to stick anything under your skin. We have a way to measure things interstitially, and the cost is way below anything else that's out there. So it could be used for third world countries, which is why we were selected out of 27 other companies that make CGM-type products to participate in a World Health Organization program that's run by FIND. And they're going to be running some of our systems in some studies in third world countries to demonstrate its cost effectiveness in those applications. We've done a lot with very little. I've raised about 3.5 million so far, and we've been able to keep that under control pretty well, and we have really moved the ball forward. We have a really good team of people that are behind me, including the ex-CEO of Dexcom, John Bird, who's the founding CEO. He's our strongest component. He's our chief technology officer. And I have a bunch of people that are really good at what they do to back me up. And we have an even better board of advisors. We are in a position that no one else exists, meaning that we can do glucose in the blood, real time, non-invasively. No one else is there. If we look at valuations, we are what we call a unicorn. The market potential is incredible. Dexcom is worth over $50 billion, and all they can do is glucose. Turns out we can do a lot more, but those are different businesses that we can pursue once we get the diabetes problem solved, because my wife has asked me, when am I going to have my device? And until she's happy, I can't look at doing anything else. So we can look at viruses, we can look at signs of Alzheimer's. There's a lot of other things we can look at. And again, this is what we're looking to raise. It's going to pay for IP, clinical trials, the whole 9 yards. At the US, we're going to be doing a type 2 device. It's going to be a PMA process, which should be fairly quick because it's non-invasive. And we have similar discussions right now here in the UK, in Germany, to do trials here. This is our Motorola brick, which is a 2-inch by 3-inch by 6-inch device. That was reduced to this size. That little green card you see there, that replaces all of that. And this is what the device is going to look like, slimmed down, but that's the device actually working on a Samsung Galaxy Watch. And with IMEC, or CCM, we'll probably develop the next silicon spectrometer on a chip, which will reduce the cost further and also increase battery life by a factor of 10. So I appreciate you being here and listening to my presentation, and hope to have your support to make this a reality and change the course of medicine.
Thank you very much. Oh, I already see a hand. Okay, we have our first question to Marc from Erica.
Have you had any interest from partners? I could see maybe some of the smartwatch companies, because I know they have even patents that talk about this kind of tech, not what you're doing exactly, but interested in those kinds of sensors, plus a number of the regular diabetes tracking companies. How are they perceiving the technology?
They're actually very interested. We've been talking with Samsung for over 3 years. They found us. They have technology scouts. We've been very careful because we want to protect our IP, but they definitely want to have a device to compete with Apple. Apple keeps saying they're going to come out with a blood glucose sensor. But the big issue that a lot of the consumable electronics companies have is that in the US at least, there's no way around, if you're going to say you do glucose, it has to be a medical device grade system. So it's not as simple as just having a number that you're not going to be held accountable before, because people are going to use this to figure out how much glucose to give them or how much insulin to give themselves, and that could be a bad thing if it's not capable of doing it. And we are talking to Medtronic, we're talking to Abbott. They're all extremely interested because this would change their business model. I mean, we're going through a major transition here from the old days where you paid for consumables, you paid for things like that. Here now, the SaaS revenue model, which people have talked to already in several other presentations, is really the way of the future. We're going to get paid for performance. That's why we won the diabetes challenge from the Blue Cross Blue Shield, because they recognized that they can actually make money with this device, and they can keep the guys that get their biggest reimbursements from our diabetics. So we keep them alive longer, they make even more money. So it's, everybody's looking at and the physicians obviously are very happy because now they can actually help manage the disease state because they have the data they need.
Okay, thank you. Next question coming from Andre Zimmermann.
Yeah, thanks for the presentation. Very impressive. Key question from my side is how many clinical data you already have and how do they compare to the classical blood glucose lab tests?
Yes, so we have 27 patients that were studied in the complete care data, which was in one of the charts here, but I can send you more information. We have a 12-person study done at complete care. And that's been the biggest criteria that we've had, is that people want more data. So the trials that we're going to be doing now that follow will be generating 160 more data points, and then the fine thing is another 80. So hopefully we'll have a lot more data. We were focusing initially on the type 2 and the borderline diabetics because nobody had a real solution to that problem, right? But when the FDA said we have to be able to do medical grade, we realized that we have to do type 1 as well. And I had a lot of type 1s that are actually mad at me saying, why aren't you doing this for us? And I said, well, you guys already no, I don't want to have this thing in my arm all the time. And so, yeah, we need to get more data. And that's what we're going to be working on. But the data we have shows it works, and it works as well as any of the other devices that are out there, any other CGM. It works as well as any of the other devices that are out there, any other CGM.
And how do these data compare to the classical blood glucose lab tests?
So YSI is what they typically use. When we for the study that we did, we actually used Roche SMBG because that was all people would have. But we also ran our own YSI, and that just reduced the margins down to even under 10%. So because of reference, it's actually easier. The actual gold standard makes the easier to make your margins because it's more accurate, right? Your precision of your reference is going to affect your overall precision of your measurement. So we see it being as good as any of the CGMs that are out there today, just based on the data we have. And I think as we get more data and we refine the technique and the AI specifically, I think we'll get better even than anything else, any of the CGMs that are available today.
Thank you. Final question from Jens Baas, TK.
I'm sorry if this question has been asked before. We have a very terrible sound quality here, so it's hard to understand. My question would be, as a payer organization, actually, we're paying a huge chunk of money every year for test strips. So it's a very interesting product for us. But if I look correctly at in your proposal, you only have a study for 27 patients. Is that right? Because that would be a number that would a little bit be of concern if your whole study is 27 patients.
Yes, and so what we always have to do is get enough data to convince people that it works, right? So we've got that. So to get the regulatory approval, we've been told by the US FDA that 80 patient study should be more than enough because we're non-invasive, but we're going to have to institute hypoglycemia, which means you have to put people at risk in a clinical setting. These trials that require that type of testing are extremely expensive because you have to have someone in a hospital, because when you put their blood sugars very low, they can wind up going into a coma, which is what almost killed my wife, or even having a seizure, which I've seen happen a few times, which is scary. So yes, we know we have to get more data, but the data we have shows it's good enough to justify that investment. What we're trying to do is de-risk it as much as we can for investors to show that this is real, and we just need to have a little bit more money to get past the finish line and do the hundreds of people. And the more data we get, the better it'll be.
I think this is a nice sentence. The more data we get, the better it will be. Thank you very much, Marc.
Thank you. Appreciate it. Thank you.
Next up, it's the same continent she's coming from. We have Margaret Magdesian from Canada, and she's going to present. Oh yeah, I hear something from the Canadian booth here. So Margaret is going to present us Ananda Devices, which is a startup launching a platform to robustly test toxicity and efficacy of compounds to over 3,000 neurons in only 30 minutes. Your stage.
I hope everyone is hearing me well. And let me check if this is working properly as well. Yeah, okay. So thank you everyone for this great opportunity. My name is Margaret Magdesian. I'm the CEO and founder of Ananda Devices. I'm a biochemist with a PhD in pharmacology and biochemistry. And I've developed a very game-changing technology where we can use to deliver safer and better medications for patients suffering from neurological diseases. I saw the opportunity of launching this technology in the market after 10 years of development, and I launched the company Ananda Devices. We have now signed contracts with multiple corporations, including 5 of the top pharmaceutical and cosmetic companies. And let's talk about the motivation. So why am I here today, and what makes me wake up in the morning? We know that 20% of the world population suffer from neurological disorders. This is 1 in 5. And the numbers are just increasing as we get older. Today, 1 in 59 children is diagnosed with autism. And as the population grows older, the numbers increase. So 1 in 5 adults is suffering from chronic pain daily or almost daily. And 1 in 3 seniors are dying from Alzheimer's or another type of dementia. For us women, the situation is even worse because we have higher chances of developing neurological disorders such as Alzheimer's and multiple sclerosis. And on the other side, we are the main caregivers because neurological diseases has a huge impact, not only for the patient, but also for everyone around them. And I know this really well because my mother suffered from a neurological disorder, and I have been next to her bedside during my whole childhood. We have been from one painful treatment to the next, and we couldn't find a cure, we couldn't find a proper diagnosis, and unfortunately, there's no effective therapy. And this makes us ask ourselves, you know, when you're there seeing all those patients, what's happening? What can we do about it? What are we exposed to during our lifetimes that is causing such a toxicity to our neuronal networks? And if there is a break in the neuronal network, and a break in the connection that is painful, that causes dementia, how can we solve it? What can we do to solve this? With this, I'm not the only one to ask those questions. Regulatory agencies have also asked the same questions, and they require that every product before launching in the market that will be in contact with humans has to be tested for toxicity and efficacy, for safety and efficacy. However, all the tests are performed in animals and not in humans. Animals are great models for cancer. I don't know if you have a dog, But if you have a dog, you know dogs can develop cancer. And cancer, if you test a medication against cancer in animals and you test in humans, usually the results are translatable. However, for neurological disorders, it doesn't work this way. You know, animals do not naturally develop autism. They do not naturally develop Alzheimer's, multiple sclerosis, and so many other diseases. As a result, animal testing for neurological diseases is very inefficient. 99.6% of all the drugs targeting neurological diseases tested in animals failed in human clinical trials. And the number one reason for all products, all pharmaceutical products rejected in the market is neuronal toxicity and lack of efficiency. It's a very slow process, takes 5 to 15 years to validate, is costly and ethically challenging. So where are we today with neurological disorders? If we're thinking about the COVID pandemic that is affecting between 5 to 10% of the population, imagine the neurological disease pandemic that affects 1 in 5 of us, and as we grow older, 1 in 3. So there is no cure, no efficient treatment for humans with neurological disease, and we all know someone, right? With the current COVID pandemic, the numbers are just increasing. We all know someone in the family who suffers from anxiety, depression, chronic pain, or dementia. And what are we doing about this? As life science professionals, we have to do something. I have dedicated my life thinking about a solution. And what I thought was, imagine how great it would be if we could grow human tissues on a chip. If we could grow neuronal networks on a chip, and you know, mini networks that precisely represent what's happening in our brain. Because that's a key issue with neurological disorders. You know, cancer, the patients free-willing give you the tumor to put under a microscope and test whatever you want. But if it's a neurological disorder, nobody's willing to give a piece of their brain. In addition, we cannot test in humans. Animals are not efficient. The main reason things are not working here, and neuroscience is at least 50 years behind, is because we do not have a proper model. So I thought, what if we could have, you know, those models on a chip? And then we could add a drug and see, oh, look, patient 1 responded really well for the drug. Patient 2, not so much. Now imagine we could do this robustly in scale with 88% reproducibility from well to well. We could image 3,000 neurons in 30 minutes. We do a multifactorial analysis and correlate with clinical, genetic, and imaging data. Well, that's exactly what I have been doing. And these are real images from our real patients. And we have rapid access to patients' response before and after exposure to compounds. I can compare my technology to, let's say, a traffic network, right? Let's image the network in Düsseldorf before and after an accident. That's what we're doing. We're imaging before and after, and we can compare the images and evaluate where the accident was and what it caused and how the traffic stopped. Today, our technology is being used by major companies to test compound toxicity preclinically, and most recently, clinically for patient stratification to define which patient will respond to therapy or not. We are adding serum from patients in our devices and see how the neurons respond to different compounds. So basically, our solution is, I brought the product. Basically, our solution is a microplate in form of products or services where we can put the neurons, the compound, we get the readers, and we get a lot of data. This is the business we're running right now, and we're looking for investors willing to invest $6 million so that we can multiply sales and add sensors and AI to our platform. We have done all of this during 2 years pandemic with everything closed, and we have a full manufacturing capacity with 2,000 units per month based in Canada. So by adding sensors and AI, we will be able to get much more data and predict how patients will respond to a certain compound. And then we can have access to a 1.7 trillion precision medicine market. How does it work? Just let's go back to the slides with the idea, right? Now we're just going to the same product, but a version 2.0 for live monitoring patients, short and long-term response to compounds. Basically, let's try to monitor the traffic network in Düsseldorf live, imaging all the time what's happening in every street. So we have the microchips, we add sensors, we grow the neurons again, and then we add the compounds. And then we can live see how those neurons are responding. Those are also real images from experiments that we are doing for specific clients. Now we get a functional data at every 10 milliseconds. Each plate of our device can image 3,000 neurons in 30 minutes. Again, with the multifactorial analysis, correlate with clinical, genetic, and imaging data. Combined with advanced analytics, our platform will be used to predict compound safety and efficacy to humans. So, summarizing, who are we and what have we been doing? So I developed this technology during 10 years, and then we launched as a product where we can grow human neurons on a chip 60 times faster than they grow in vivo. We signed contracts with the major corporations that are now recurrent buyers from our products. Next, we signed co-marketing and distribution agreements during the pandemic with other partners that are using our products as well, and we're targeting the R&D market, which is a $2 billion for pharmaceutical and cosmetic companies. We're also working for the regulatory homologation of our devices, especially for an in vitro diagnostics for specific treatments in partnership with some of our customers. And we're now trying to partner with, actually we already received the contract. We're partnering with the major sensor producer in the US to add sensors to our platform and target the precision medicine market. Why us? We're a proven team. We're a multidisciplinary team. We have 7 PhDs in engineering, neuroscience, cell biology, and biochemistry. We have an IP portfolio. We have customers and recurrent customers. We have recurrent investors that came back to us. We have established processes and certification and reputation with financial agencies and especially with our customers. And we won 24 awards in science and innovation, including the best technology for laboratory automation, L'Oreal in digital, Bayer Crop Science, Cartier Women's Initiative, and many others. But most important, we don't only have the technology, we don't only have the clients and the financing, we also have the heart. And we are interested in helping patients get a better diagnostics and better outcomes. Thank you.
Thank you very much. Do we have questions for Margaret? Angus?
Margaret, I noted that you cite revenue. Can you give us a sense of how much in revenue and what your projections are for the next couple of years?
Yeah, so we have recurrent revenues, and we are now working on a pipeline of $3.8 million.
That was a quick answer.
Yeah,
we have another question from Rainer Christine.
Sorry.
Margaret, I noted that you cite revenue. Can you give us a sense of how much in revenue and what your projections are for the next couple of years?
So we have recurrent revenues, and we are now working on a pipeline of $3.8 million.
That was a quick answer.
Yes. Elaborating it very well. Thanks very much for the presentation. Could you comment on the competitive landscape a little bit? I guess the challenge is pretty clear and the relevance of what you're doing. And to my knowledge, there are quite a few companies pursuing the topic in general. It'd be great to understand your view, how you are different, who you feel are your closest competitors along those lines. That would be really helpful to understand.
Sure. So our main difference are 3. Number 1, physiological relevance. Number 2, time. And number 3, scale. So we are the only technology that I know that can organize neurons exactly like they are organized in the brain. The only similar thing I could see is, like, we're building neuronal networks, reproducible neuronal networks. It's not just an entangled mix of neurons and not what they called organoids, mini brains, or something that is just a centrifuge cells on a ball. All our networks, we have 88% reproducibility from well to well. In addition, our platform is a microplate that is, I don't know if you guys can see this, that is compatible with all standard readers and standard workflow. So people can image 3,000 neurons without buying a full new equipment or anything. It's in 30 minutes you get the results. Also very different from what's in the market, which are mainly, much larger platforms where people have to buy a new lab to make sure it fits and they can scan a certain amount. Besides, because all of our neurons are well aligned, it's a matter of imaging recognition. Like if you see an image, which is today very common in AI, right? You can compare images really quickly. Our neurons are like a barcode in each of the wells. So every small difference from one to the other, it's quickly analyzed, and we deliver the results in less than a day.
Thank you. Do we have other questions for Margaret? Not in here on site. Do we have some other virtual questions coming up? No? Okay, then thank you very much.
Thank you.
And we can move on to our next finalist, who is Ignasi Belda, pitching us MiWEndo from Spain. And MiWEndo is presenting their technology, which is an early colorectal cancer diagnostics disposable medical device.
Well, thank you very much, and thank you for the honor of being here today with all of you. As it has been introduced, I'm Ignasi Belda, and I'm the CEO of MiWEndo Solutions. Let me see how it works. Okay. As you probably know, colorectal cancer is one of the main health burdens in the world. There are many countries, as it happens in Spain, but not only, that colorectal cancer is the most common cancer in that area. In fact, every year in the world, there are almost 2 million new cases. And unfortunately, the survival rate, the 5-year survival rate, is only of 50%. The reason for such figures is how the gold standard for diagnosing colorectal cancer works. This gold standard, as you probably know, is colonoscopy. And colonoscopy, unfortunately, is not very efficient. Actually, 22% of the polyps in a regular exploration are never seen, which means that 8% of colorectal cancer come from a negative colonoscopy. That means patients that took a colonoscopy, they were told that they were okay, and unfortunately there were their precancerous lesions, and some months after, then they develop a cancer, and this is called interval colorectal cancer, which is 8%. Okay. To solve this, we have been working for many years in a new medical device. Actually, I have here a sample. It's pretty small. If you want, later in the coffee break, you can hold it in your hand. And this medical device is an accessory that we have designed to attach at the distal end of the colonoscope. As you can see here, the white tip on the distal end of the colonoscope, right? So it's a single-use accessory that you put on the distal end of the colonoscope, sorry. So every time that you are performing a new colonoscopy, then you have this accessory which implements a radar. So this radar uses a very disruptive technology called microwave imaging and provides a 360 degrees of detection, right? So without changing the medical practice and with a very safe radiation, we have designed this low-cost device that can be used in any colonoscopic exploration. So during the last months, we have been working in validating our technology with preclinical data. First, we did some studies with ex vivo human samples, and we achieved 100% of sensibility and 100% of specificity. Then we are working with porcine models to test safety. So we are now finishing these experiments with the porcs, but according to the results that we have already, we can demonstrate no adverse events, and very important, we can demonstrate that our device does not affect the maneuverability of the colonoscope. So this is our market. We are in a market of 20 million colonoscopies per year in our 7 target markets, which are Germany, UK, Italy, France, Spain, US, and Japan. In these 7 markets, where our patent is filed, there are almost 17,000 hospitals doing colonoscopies every day, and this means an annual market of more than 1.2 billion euros. This is our current roadmap of our development. Now we are finishing the experiments with the porks, and in the next weeks we will finish these experiments, and then we will start filing all the authorizations to start our clinical validations, which will start in April 2022, using 65 patients. And with these results, we expect to have the CE mark, and afterwards we will start a second trial for marketing purposes in 2023 with almost 1,000 patients. And all this has been financed with 4.5 million euros that we raised from private and public funds. And for the marketing studies, for marketing clinical studies, we are raising 2 million euros and we will start next month to do this. So this is our competitive landscape. As you can see, there are some other technologies in the market that are trying to improve the performance of colonoscopy. Our technology compares to the others in 3 different ways. First of all, we provide, as I was saying before, 360 degrees of detection. In addition, our technology also helps to automatize the detection of polyps. So now, in some way, we can democratize the high performance of doctors doing colonoscopy explorations. And finally, according to our preclinical results, we hypothesize to have a 20% of increase in the adenoma detection rate, which is the gold standard for comparing all technologies in this field. Our business model is to sell these accessories, single-use, disposable, at the cost of 120 euros via B2B distributors, and our first sales are expected to start in 2024. Then, according to our financial models, the payback period of our project will be 3.5 years after first sales. However, we have to admit that this is a very hot market of mergers and acquisitions. And here you have some examples of the recent year. For instance, Boston Scientific, a company that we are discussing with them very tightly, they acquired another company in the field some years ago for $210 million, or Ambu acquired in 2017 MiWEndo Medical for 225 million euros, etc. Finally, well, this is our team. Actually, nowadays we are 12 people because we incorporated 3 more people in the last weeks. And this is the list of our international partners where we are planning our clinical studies here in Germany, the Netherlands, Belgium, Spain, of course, Portugal, etc. Well, thank you very much for your attention.
Thank you very much. Do we have questions for Ignasi? Erika, coming for you.
How do you think about the Cologuard technology and some of the molecular screening technologies for cancer? And do you think of this as, I guess the question is colonoscopy a durable market at its current size, or do we expect it might shrink?
Yeah, this is a very interesting question because there are now in the market many technologies that are trying to diagnose colorectal cancer with liquid biopsies, for instance. For us, this is very interesting because we believe that thanks to all these technologies, the number of colonoscopies will increase because, believe it or not, colonoscopy is the unique way to remove polyps if you have a polyp, if you have a cancer, right? So the more people is screened with new technologies that are low cost and non-invasive, etc., the more colonoscopies we will have in the hospitals to remove the polyps and the cancers that have been detected. So for us, it's a really great news, this new emerging market of liquid biopsy that detects with very few sample of blood new cancers. Thank you.
Thank you. Bernd Görgen, HTGF.
Yeah, thank you. If you sell your item as an accessory item to attach it to the endoscope of a different manufacturer, did you already get in touch with Olympus or the other guys in order to get their permission to attach your technology to their endoscopes? Otherwise, it wouldn't work, right?
Sorry, I didn't understand your question from here. I cannot listen
Yeah,
very
I'll
well.
repeat my question. Did you already get the official written permission of one of the endoscope manufacturers in order to be allowed to attach your technology to their endoscopes?
Yeah, we have been discussing, I don't know where to look to. We have been discussing with 5 of the 10 manufacturers of colonoscopes in the world, Olympus, Fujifilm, Medtronic, Boston Scientific, etc., Ambu, for instance. And well, the first thing to clarify is that we don't need permission from them to attach anything, any accessory to their colonoscope. Nevertheless, they are very interested, and they are all of them following up very, very tightly, because what they really would like to see is how they can integrate our radar inside their tube, inside their colonoscope, right? So they add more value to their colonoscopes and provide more tools to diagnose colorectal cancer rather than optics, because nowadays mainly is colonoscopes are optic tools.
Happy with that answer? It seems. Good, then we can move on to Jens Baas, TK.
Well, thank you very much for your fascinating presentation. Being a physician, I performed a couple of hundred colonoscopies myself, so I'm naturally fascinated by your product. But I have 2 questions. First of all, it's a single-use and throwaway product. So how is it working on a technical basis? Is it like beeping if it's coming close to a tumor, or what's it actually doing? It's not transmitting a picture, obviously. So what is it doing if it comes near a polyp that it's suspected to be malignant? That's the first question. And the second question is the price point. Price point of 120 euros means the colonoscopy would be roughly like 20% more expensive to reimburse than it is right now. Are you not afraid that that is actually too high of a price point to get a bigger market penetration?
Yeah, well, thank you very much for both questions. First question is very interesting, actually, because I forgot to explain that, yeah, as you were imagining, this is giving an acoustic alarm if we detect a polyp. So we put that on the distal end of the colonoscope, so when you pass next to a polyp, then the medical doctor that is performing the colonoscopy is listening a kind of beep, like beep, 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 like the car when you're parking backwards, and then you are close to the wall, so something very similar. Okay, that's the first question. Second question, with respect to reimbursement, we have been doing a market study for reimbursement proposal, market access study in the 7 countries where our patent is filed. So we did the study based on 2 parameters. One is the increase of polyps, or polyp detection in colonoscopies. As you were saying, if we go via this pillar, then we are increasing enormously the cost of the colonoscopy. Well, not enormously, but in a significant way, the cost of the colonoscopy, and we don't think this is the good strategy to look for reimbursement. The second strategy is the strategy of reducing colorectal cancers in the system. And then, according to this strategy, we did the studies in these 7 countries, and for instance, in the US, the cost of our device could rise up to $600, which is not the case, because we want to keep in the range of $100 or 120 euros. And it's pretty same in Germany. In Germany, our device will support something like 500 euros per colonoscopy.
Okay, do we have other questions on site? Doesn't seem like it. Thank you very much.
Thank you very much.
Next up, we have Irubis. They call themselves the future of biopharma 4.0. And with their product Munipa, Irubis is optimizing the monitoring of critical process parameters and manufacturing processes of the biopharmaceutical industry.
On average, it costs more than 1 billion euro and takes more than 10 years to bring a new biological drug to the market. A significant part of these costs arise in manufacturing due to the lack of automation. But the faster the production batch is released, the faster it can be sold and the faster patients can obtain the life-saving medications. But how to accelerate drug production? So I'm Anja from Irubis, and we automate bioprocessing by real-time monitoring and control of critical parameters. So in cell culture or cell cultivation, the so-called upstream process, but also in protein purification, the downstream process, there's still a huge lack of automation. So this means they take every day manually a sample out of the bioreactor, put it to a separate device and analyze it. This costs high process development costs, no real-time monitoring, a low degree of automation, and also a high contamination risk. And we changed this with MoniPar. So MoniPar is the first spectroscopic plug-and-play device for continuous monitoring. Plug-and-play in this case means that if you have other spectroscopic systems, it takes up to 6 months just to calibrate the system to the certain cell culture, but also to the media. And we can reduce this time to 1 minute. So it's really plug-and-play. And on the other hand, we also have a single-use component, which is highly important in the biopharmaceutical field, as we can avoid contamination. And also by this external flow cell, we also have a constant measurement environment, so we can also get better results. So how does this innovation work? So as glucose control is our entry market, you can see here a bioreactor, which is connected by a loop to our instrument. And we pump the fluid 24/7 through it. And with MoniPar, we evaluate the glucose value. And if the concentration is too low, we control the pump to feed the glucose level. So no manual testing is anymore necessary, and the glucose concentration can be kept on the perfect level. But why is it so important to control glucose? So on the one hand, of course, if you have an automated system, you have no biologists that need to come on the weekend, so you can save operators' time, but you can also save lab equipment maintenance costs. The more interesting point is that a constant glucose level or a perfect glucose level can lead to a product yield increase of up to 60%. This means that it's a benefit of up to $9 million per year. And single use is so important, as in manufacturing, contamination is one of the main problems for batch failure. So if you take, for example, a blockbuster drug, a month production stop can result in a lost revenue of up to $1 billion per month. So we are planning to sell MoniPar for $150K, but we also have recurring revenues as our flow cells are single use, and we also can sell some software upgrades, as in the spectroscopic data also further parameters are already included. So the main markets are Western Europe, US and China, and our sales profit at one biopharma company has up to 100 bioreactors. So it means we can sell to them up to 20 MoniPars. Then they also have different departments, so we want to automate the whole process. So we can also have some kind of cross-selling there, some upselling to different locations, and we see further markets also in mRNA, cell and gene therapy, and also in tissue engineering. So we assume an addressable market of in 2027 of 4 billion euros, but we also have further markets in the whole bioprocessing. Of course, there are also other peoples in this field. So far, there is no real-time monitoring as widespread or is widespread in this field, and we assume that only 1% of the processes are monitored. So the main competitors are in the field of Raman spectroscopy, but they still suffer from high cost and also low degree of robustness. And I think the most important one is that it takes a lot of time just to calibrate the system to each cell culture media, and this is not acceptable for the customer. And by realizing a one-point calibration and then plug-and-play device, it's a real game changer in this field because you can use it directly. So we are the co-founders, that's Lawrence, Alex, and me. We founded the company in 2017, and in our team we combine more than 2 decades of experience in infrared spectroscopy and are authors of 15 publications in this field. And one of our biggest success regarding fundings was last year that we won the Horizon 2020 award, which helped us to bring MoniPar to the next level. So in the last year, we had a close partnership with Amgen to develop MoniPar, and we also had 6 other paying biopharma customers to get all the customer insights to fit our product to their needs. And so we are seeking for 2 to 4 million euros. And now I'm looking forward to your questions.
Thank you very much. Oh, we have a juror we haven't had today. I'm gonna give that word to our DxPx initiator, Mirko Stange.
Can you tell us a little bit about your patent portfolio, what is protected, and with this regard, can you also measure other things than glucose? What's in the pipeline?
I'm sorry, I missed the...
Can you measure other things than glucose levels? And if so, what is in the pipeline and what is your patent protection?
So I just gave glucose as an example as it is the easiest one and as it is a control parameter, but we also can measure lactate, and it's also possible to measure ammonium, glutamate, glutamine, so almost all metabolites and nutrients in a bioreactor, but it's also with infrared spectroscopy possible to measure protein quality and quantity in downstream processing. So it's a large field of different parameters which are interesting. Yes, so we have 1 patent which is granted, 1 which is in application, and 2 we are working on.
Other questions from our jurors? None. Okay, then thank you very much. So 7 down, 1 to go. Last but not least, we have Daniel Fallscheer. He's presenting DasLab. DasLab is a startup connecting labs, sample takers, clinicians, and logistic providers to enable scalable and reliable delivery of at-home and professionally sampled diagnostic tests throughout Europe.
Thank you. I couldn't have summarized better. Thanks for that. So I'm Daniel, co-founder, CEO of DasLab. And how should I say, I'm impressed always to see a lot of real devices here in those presentations. What you're now going to hear is really an infrastructure play, a digital infrastructure play in the lab diagnostic space. So what is DasLab? DasLab is a digital lab test infrastructure. And we think about interoperability in an ecosystem. So we think about healthcare as an evolving ecosystem. So we think about connectedness. And at the end, what motivates us also as a vision is really medical outcome. We want to improve medical outcome. We want to get healthier patients by more easy and universal access to lab testing. And what's the mission? By 2025, we want to be the leading provider of testing, agnostic and neutral testing in Europe, at home, but also professional. And guess what? The company is just 13 months old. It was not invented because of COVID, but COVID was, so to say, a little bit of an unfair advantage for us. Because suddenly what we realized in COVID is that there is a big lag of digitalization of end-to-end workflows when it comes to testing. So accessibility, booking, finding locations, integrating with labs, but ultimately also having the consumer control over their results, getting travel certificates and so on. And what we found on top of that, and I think for the informed audience in the room and in the call, I think this is not a big surprise, that there is a lack of, I would say, deployment of data standards. Those are there. FHIR is there, LOINC is there, SNOMED is there. But are they deployed already in a broad scale, in this ecosystem? No, they're not. And you have to go to every single lab location sometimes, plug yourself in, and you realize it's not an easy to plug-and-play solution and not easy to share data across those sectors. So the problem is also, from a data perspective, it's outdated. Still quite a lot of paper involved, still a lot of fax machines involved, still a lot of sending out even PCR results in COVID still today as we speak via post. And that takes sometimes 2 days to receive the result. This is simply not acceptable, I guess. And on top, what you also see, as I said, data standards not fully deployed in a very stringent way across Europe. And we are at the moment active in Germany and Spain. It was a deliberate choice. It was not by accident. We love really Spain also. The market is great. But definitely other markets in Europe have the same issue, have the same fragmentation. Although consolidation, of course, in the lab space made a big progress in the last maybe decade, still, if you dig deep into this integration with those labs, it's still very highly fragmented. And it's complex. And we, from the founders team, and I come to that later, we love complexity. We don't think of simple problems. We love complexity operating in a complex health ecosystem, and we love to be a partner with existing providers in that game. So what is DasLab at the end? DasLab is at the end, first of all, an infrastructure which is digitally accessible. So that means we're connecting labs, we're collecting sample takers, logistics providers. And for whom are we doing that? For providers. So the first providers, the first ones that will be going live after COVID, at the moment we're doing a lot of COVID, is telehealth. We have roughly 50 opportunities just in that space at the moment across Europe. And what is their big need? Countrywide access to testing, a lot at home, but also getting access to professional sample taking. And in Germany, it's not easy. Also in Spain, it's not easy in a digital way, because still a lot of paper is involved, phone calls, and this is, I think, where we can make a big contribution to that. And it's a very simple plug-and-play solution. And what is important in an ecosystem going forward, we believe it's about standardization of the data piece. That means the results that the providers get back from us are harmonized. They get them back via FHIR, LOINC, and SNOMED. And irrespective of what the lab has, we translate this in ours based on a mapping, and labs are already quite used to that exercise. So as I said, at the beginning, it's telehealth. It's also symptom checkers. We realize also drugstores in Germany, Drogerien want to go into that field of offering additional health services and testing. I think what COVID did, testing became somewhat more, more public, more used to the people, to the broader audience, to the broader population. But at the end, if you think it through, do we add value beyond telemedicine? Yes, we do, because still we have a very sectoral healthcare system. That means those sectors are not very well connected with each other. Think about how you as a patient have to take your test results maybe from one sector, from your GP or maybe cardiologist into the hospital. Is that all connected? Can you simply, take your results with you? Is any intelligence taken out of this longitudinality of data on your personal level? No, it's not, because the issue is a bigger one, because first of all, you don't have this connectedness so easily, and you don't have, at the moment, a big standard. So that's where we're going to add value going forward. And of course, if it's about longitudinality of data, of course, at a certain point, we're going to add then intelligence on it, AI and stuff. It's a bit too early, but this one is definitely the fundament to build this infrastructure. I think we have a quite strong founding team. Next to me, I would say maybe 50, 60 years of experience in healthcare, all from big corporate experience, and all now love to be an entrepreneur and very much digital footprint. We're also not shy in doing any medical device related stuff. Our VP product, we did it in our last firm, digital medical device 2A, so this is also something where we are not shy on. And also what we don't show on that slide, but we have a quite senior advisory team consisting of former C-levels of big lab chains of Europe, or VPs of also lab chains, and also from the former, for example, Chief Commercial Office of Babylon. So all those people help us drive that business. Up until today, 500,000 tests performed after 13 months, more than 2 million revenues, currently raising a seed round, and I think we're close to closing that, hopefully end of this week, and then going for a really progressed Series A next year, because at the moment the demand that we're seeing in those markets is just massive. We cannot, I think we can easily add 5-10 business people on it to just work with the opportunities. We do it in a very focused way, prove that also beyond COVID we're going to have really a value add solution, but I think then it's a first mover advantage in Europe, and this is also, we definitely want to stick with Europe. And for Europe, we want to be this is not a quote from me, it came from really from investors and also from a founder of a big leading telehealth provider. They see us as this tribe of diagnostics. The other one is maybe really bold. Bold is also value for us as a company, but DasLab is building the largest lab chain, maybe in the world, let's see, but without CapEx. Thanks for that.
Thank you very much. Do we have questions? Not on site. Maybe if I could.
There is one in the audience.
In the audience. Maybe it would be good if I first can have a look at our jurors if someone is asking questions.
All cameras are off.
All cameras off. No raising hands. Okay, so, of course. We have one question here, okay. And maybe we cannot go after that in the audience.
So in your last slide, you said without CapEx. Is this are you planning on also developing own products or own solutions that are proprietary to DasLab?
So just to clarify, you mean, so to say, own tests or own labs or something? No, this is not our target. Our target is clearly we want to partner up with relevant, innovative players in that field. As we do at the moment, for home testing, we see some solutions which we, from an experience perspective, don't like that much. That's why we go to Medica tomorrow, meet people, we're really driving this, but always together with partners. We're a big fan to do this with smaller, family-owned, mid-sized labs, because they're very innovative and want to co-develop, but definitely our core of the company is digital. But we have the scientific knowledge, we have the medical knowledge, because otherwise we cannot speak on eye level with the medical people on the other side, definitely. But you could argue what happens if we become really big. Suddenly at the moment, we're plugging into an existing infrastructure. Is the capacity enough or not? I would say it's too early to judge what will happen in the mid to long run, but that's to be seen. But the clear vision is no. I think there was one here in the back.
Yeah, so then I think it might be a good thing to close this one with a question from the audience.
My question is related to your competitor. Do you know any of them?
Yeah, well, the competitor, so the way, as we heard earlier, we love metrics, me as well. So if you take a matrix, and basically the two coordinates are, direct to consumer, so to say, close to that. A lot at home, there you're going to find plenty players. You find, the Thrivers, the Everly Wells, in Germany, the Sarah Screens and the Lycons of the world. This is all, direct to consumer. They want to have their own brand. They want to often build a supplement business on top. This is not how we see ourselves. We want to play really in proper medical care. Then that means, we have to cover a broad range of tests, and also professionally sampled. There are in US, some companies look at IX Layer, look maybe at Butterfly Labs, look at Precise, MDX. These are companies evolving. Interestingly, US is not ahead here. I would say, this is not a copycat, really. We just found them underway. But in Europe, we are not aware that this is something similar in a vendor-neutral, agnostic way. Good?
Oh, good. Thank you.
Thank you.
Okay, so with DasLab, we had our 8 finalists up on this stage. I hope all of you enjoyed the great pitches we had. They did their job. Now we need to do our job and put out our calculators and see who got the most points from our judges virtually joining as well as on site. So we will announce the winners tonight at the reception. And I'm really excited to see who's going to win our first ever given out 42PLUS1 trophy. Yeah, I think with that I will close this pitch award. But there's one thing left you can do. You can rate your favorite finalist from this day. You simply need to go to www.menti.com and put in the code 26423201, or yeah, I see some of you are already picking out your smartphones. You can just scan this QR code and you can rate for your favorite finalist. So we will also have a publicity award winner. Let's see if those match or if we have 2 winners tonight. With that, thank you very much. Enjoy the rest of the program. Enjoy the rest of the conference, and I hope to see you all in some constellations on some tables and have a chat. Thank you.